Welcome to another exciting educational video brought to you by Vision Concepts Academy. So in today's video, we're looking at biology and you have cell structure and functions. We will see key definition concepts using simple language. What is a cell? What are the types of cell? What are the types of organism? We'll look at the structure of a typical cell for plant and animals. We'll differentiate between plant and animal cell, and we'll wrap up with important experiments done by WAEC and JAMB Insights. We'll observe animal cells and we'll do some drawings, plant and animal cells. So sit tight as we present to you what we have today. If you've not subscribed to our channel, kindly subscribe to our channel to get more content on the STEMs, physics, chemistry, biology, Vision Concepts Academy. So we'll dive in. So we start. What is a cell? So we say that a cell is the basic structural, functional, and biological unit of all living organisms. Every living thing is made up of cells. So it's the basic units. If by the time you try to divide the organism into different parts, you will end up with the basic part, which is the cell. The cell contains everything that a living thing should have, everything that a living thing should have in terms of structure, in terms of functions, and in terms of being known as the biological unit of a living thing. Any other thing beyond cell is nothing. You don't have anything living. If you destroy the cell, you have destroyed the whole living organism. Okay, so we'll go to the types of cells. So we have two types of cells. We have the prokaryotic cells and the eukaryotic cells. So the prokaryotic cells are simple cells without a nucleus. We'll look at what cell breakdown is, or membrane bound organelles. So this is a prokaryotic cell, cells without nucleus or membrane bound organelles. But eukaryotic cells are complex cells. They are complex cells. The difference, this is simple, this is complex. They are complex cells with a well-defined nucleus and organelle. Okay, so example for this one is plants. Example, e.g., you have plants, animals. These are complex, so they have cells, eukaryotic cells. You have fungal, and I can also put protists. We'll see this even as we progress in this course. So we will go to the types of organisms. We have the unicellular organisms and we have the multicellular organisms. So we'll go into the types of organisms again. Like I said, you have unicellular organisms. So these are organisms that are made up of only one cell. An example is amoeba and paramecium, just one cell. And the multicellular organism, from the name multicellular, means that it is made up of many cells. They are organized into tissues and organs. Okay, examples are humans, animals, maize, etc. Okay, so we'll look at the structure of a typical plant and animal cell. We'll dissect the different parts and components, the name of the parts of the cell, and what's the function. What's the function? Okay. So we are breaking down the structure of typical cell, plants and animal. For if you take a particular cell, these are the different organelles that you will see, different parts of that cell, and then we are going to be looking at the functions side by side. So the first one we have here is the cell membrane. The cell membrane controls entry and exit of substances. So the cell actually interacts with its environment, and The cell membrane is like a door where the substance can enter. It's just like a skin around the cell, a skin that just defines the area that the cell is covering. So it controls the entry of and exit of substances. It takes in substance, and from the membrane, substance can go out. The next organelle of the cell is the cytoplasm. The cytoplasm is like the jelly-like substance where reaction occur. So it's just a part of the cell. Cytoplasm is jelly-like substance where reaction occur. Now the nucleus is the central part of the cell, where the heartbeat of the cell is, where the DNA of the cell is. So it controls cell activities, like the brain. It controls cell activities and contains DNA. So that unique feature of that cell is what is present inside the nucleus. Okay. The fourth organelle cell part is mitochondria. Is the powerhouse of the cell that produces energy. Just like the generator of the cell, the one that makes the cell to be active. So the powerhouse, mitochondria. Please note this. Then you have the ribosomes. That is where protein is synthesized. Where protein is synthesized in the cell. Okay, so we still have more. We'll go. So for the rest part, I have endoplasmic rectum, ER. Endoplasmic rectum is functioning as transport materials. So for the smooth ER, you have fats, and the rough ER, you have protein. So that's how you differentiate the two of them. The smooth ER, endoplasmic rectum is fatty, and the rough ER is proteinous. Okay, so Golgi bodies is packages and transport materials. So Golgi bodies packages and transport materials. It's like the nerve, it's like the bloodstream. So it package and transport material. While the vacuole is just a large space that stores food, water, waste. In plants, you see that the vacuole is larger in plants. So if you see two cell samples and one vacuole is larger than the other, definitely it's also that it's for plant. Of course, we will see some other features that are only attributed to plant cells, like the cell walls are for plants. The cell walls for plants are defined and structured, something like this. So these are cell walls for plants. They are defined and structured. It provides shape, you understand? Animal cells are mostly shapeless, you understand? But for plant cell, you see that it's rigid and has shape. The cell wall for plant is responsible for that. So we have the chloroplast. The chloroplast is also for plant only, and for chloroplast is the site where photosynthesis occur. Okay, so all the cells in the plant having chloroplast is where the photosynthesis occur. And then finally, I have the lysosomes. The lysosomes are the digestive waste materials. Only animals have this. Is where the digestive waste materials happen. So we're going to be looking at the differences between the plant cells and animal cells. Okay, we'll look at the differences. Like for plants, cell walls are present and chloroplasts are also present. Okay, so in a tabular form, I have the difference between plant cell and animal cell. Basically, we'll be looking at features, what is present in the plant cell and not present in the animal cell. So the first I have in the list is cell wall. The cell wall is present in the plant cell. Recall, I told you that this cell wall is what makes it regular, to have a defined shape. Cell wall is absent in the plant cell. Instead, what you have there is membrane, something that covers, just defines the outline of that animal cell. So for the chloroplast, when you see chloroplast, what comes to your mind is photosynthesis. Understand? And you know only plants that undergo photosynthesis. So chloroplast is present in the plant cell, but absent in the animal cell. So that's another difference. The third difference is the vacuole. The vacuole is very large and central in the plant cell. Large. Recall I told you that the vacuole is where food substances are stored, provide like storage for food, water, and waste. Stand. And it is small or absent in animal cell. They don't need the vacuole for that such function. And finally, last feature I have here that would have a difference is the shape. The shape of the animal cell, the plant cell, is regular and rectangular, while that of the animal cell is irregular and roundish. Okay, so we're going to be doing like important experiments in the biology lab to, one, observe plant cells, and to observe animal cells. I just segmented each of two so that you can know the difference. So if I want to make an observation on plant cells, for instance, the steps I'm going to take is, number one, I would, for example, peel an onion layer. Just take one layer of the onion, peel it. Then mount on a slide and stay with iodine. So I'll put it on the slide and stay with iodine, and view under microscope. Now, when I do that, what I will observe is that I will see the nucleus, that's the central part of the cell, where the DNA is resident. I will see the cytoplasm, where photosynthesis occurs. I will see the cell wall, that is the regular shaped cell wall, and the vacuole, where you store food, water, and waste. You will see all these things. We have described them before. So, but how to observe it, that's what I'm explaining to you now. Then for an animal cell, when you swab, the steps you take is to swab cheek cells. Swab cheek cell. What is swabbing of cheek cell? So swab cheek cells refers to a simple biological process of collecting cells from this inside of the person's cheek. So you can use a sterilized cotton swab or stapler. Just use a sterilized cotton swab to collect just a small sample. Understand? It is commonly used in the biology lab or classroom to observe human cells under the microscope. So that's what you do. Just how it's done. Just gently rub a clean cotton swab or toothpick on the side of the lining of your cheek. The swab picks up cheek cells. You smell it on the microscope slide. Smell means that you just put it on the microscope slide. Okay. So you now, the next thing is to do is to stain with metal blue reagents. Then we'll now view under the microscope. So when you view under the microscope, what you observe is that you will see the nucleus. The nucleus is the central part. You see the cytoplasm. You will see the cell membrane, and they are all visible. Okay. Sorry, I think I was saying this is for photosynthesis. Actually, the chloroplast is for photosynthesis. Please, it's the chloroplast. All right. So let's go into a practical diagram of these cells and identify these parts in the cell. Okay, so we have a diagram showing the plant and the animal cell on the board. You will see that the diagram is drawn and multiple features should be present. For any plant cell, let it have a distinct four-corner rectangular-like shape. You have the cell wall, the cell membrane. We'll define all these parameters, all these organelles, Golgi apparatus. You have the vacuole, you have the chloroplast, you have the mitochondria, you have the nucleus and the nucleus, you have the ribosomes for plant cell, and the Golgi bodies. Okay, the cytoplasm, the chloroplast is for photosynthesis. Okay, and then for the animal cell, it should be circular or shapeless. Golgi apparatus is there, nucleus is there, centrulose is there, cytoplasm is there, ribosome is there. You have the rough ER, endoplasmic reticulum, and the smooth ER. We also have it here too, but it's not there. The key thing in your exam is when you're asked to draw this, at least for the WAEC exams, make sure you mention at least 5 of these organelles. Make sure you mention at least 5. And your diagram, let it be neat. Let the arrow go out and you write the name beside the arrow. Don't cross arrow like this, and don't write name like this. Don't write name like this, because your examiner needs to see what you are doing. Let it be neat. Let it come out, from you draw a line straight and write it neatly. Okay, you have to do a better job than this. This is just presentation. So write neatly to show distinctly what the organelle is. So, like I said, make sure you do at least 5 labeling for them to know what you are actually doing. All right. So, I will leave some questions in the description and the comment section, and subscribe if you're not subscribed. I hope this video has been informative for you. I'd like to thank you for viewing.